Okay, I'm going to start talking about the chemical senses today, and I plan to finish up talking about the taste system and taste, and I hope to at least get somewhat into olfaction, or our sense of smell. I do suggest that, so this is usually my favorite time of class as I start to do these in-class laboratories or really demonstrations, and sometimes at the end of a demonstration, someone says, "What was the point? What was I supposed to learn from that?" Even though I feel like I'm saying it. People don't necessarily catch everything that I say. And the way the PowerPoints are this semester, it's kind of nice that you can go back and maybe say, this is where she talked about doing this. I actually did this demonstration for myself, and I can go back and listen to that PowerPoint just for that one slide again and get the point of why, what was I supposed to learn from this? Our chemical senses are really considered the gatekeepers, okay, that they are keeping us safe to a great extent, because when we are tasting something, we are taking that environmental stimulus itself into our body. And so it can either be safe or unsafe. And in the safe category, it can either be good for us or not really all that good for us, not really giving us much of anything. So what taste gives us is it lets us know which foods to eat and which foods to avoid. And so this is based on the quality of taste and its effect. Throughout evolutionary history, we have not had enough food many times in our evolutionary history, and other animals are in that same situation. And so for humans, we like sweet compounds, and we also like things that tell us that they have some kind of fat, because fat gives us fat to get us through the winter, and sweets give us some kind of energy. And so when we eat sweet compounds, we have this automatic acceptance response, and it prepares the gastrointestinal system for glucose and for something that is going to fuel the body. This is actually one of the problems with things like aspartame and artificial sweeteners, is that it tastes sweet. We're having this preparation of our gastrointestinal system as if we're going to get some kind of glucose for energy for our body, for our brain. If I haven't said it in this class enough, everything uses glucose. Our neurons use glucose in order to work. Everything uses glucose. And so if we're preparing for that and yet it's not actually coming because we're eating an artificial sweetener, then we're going to be more hungry and crave sweets more because of that interaction. We have bitter compounds where we have an automatic rejection response. Many of the poisons in the world are bitter. So strychnine, arsenic, cyanide are all bitter compounds. And oftentimes bitter tells us that something is poisonous and bad for us. We also have salty compounds, which tells us about the presence of sodium. We may crave sodium if our body needs sodium. So there are times when we are sweating out sodium, when we are requiring sodium, and we are craving sodium. The connection between taste and our bodily response is not always perfect. So my usual example is sweet compounds, which throw us off, those artificial sweeteners, but also with bitter compounds. Many of our bitter vegetables are obviously not poisonous, but they are extremely good for us. As I said early in the class, it is those green leafy vegetables that are nourishing our pigment epithelium and various parts of our brain and our sensory systems. They are really good for us. So this connection between what we know about taste and how the body is going to respond to those compounds, that's not a perfect connection. Much of our research on taste and different taste qualities have been done on 4 basic taste qualities, including salty, sour, sweet, and bitter. And so in the parentheses there, I have a really good example of each of these that are, the examples are specific to that taste quality. So sodium chloride tastes salty, hydrochloric acid tastes sour, sucrose tastes sweet, and quinine tastes bitter. Over the last, so I think it was in the 1990s that people suggested that we might have, or it looks like we have, receptors that are responding to umami. A good example of an umami taste is monosodium glutamate, or MSG. It is sometimes described as savory or meaty. Okay, so I've mentioned to people that I get tired of my own cooking. I'm a vegetarian, and they go, "You're missing the umami." And I thought they were talking about my mommy's cooking. I was like, but no, they meant umami. I'm missing that savory kind of that whole taste quality. Don't think that actually explains why I'm tired of my cooking a lot, because I tend to cook the same meals, but nevertheless. So that's a fifth basic taste quality that we've discovered, although most of the research that we're going to discuss, because throughout history they talked about those first four, but we also have added, even more recently, it looks like we have receptors for fatty acids and fats, and they have suggested that maybe we should call this oleogustus or oleogustus. Not sure how you say it, not sure it's going to catch on. There is no real consensus yet as to whether or not we should consider ourselves to have a receptor for this and what we should call it. But as I mentioned before, the things that we've needed evolutionarily throughout our evolutionary history are glucose, so knowing that something's sweet, and knowing that something has something that gives us fat, and those fatty acids we tend to crave fats sometimes as well, which suggests is one piece of evidence that we have taste receptors, that this is a basic taste quality. But actually, the way we examine whether or not we have taste receptors is through adaptation studies and cross-adaptation studies, and it looks like, again, researchers are finding that yes, we have some kind of receptors for fats or fatty acids. Even less research has been done on those, because that was in sometime in the mid 2000s, before 2010 is my memory of when they proposed fats or fatty acids as another basic taste quality. So mostly I'm going to be talking about those first four. And what you can see over to the right are some magnitude estimates. So the first one is a magnitude estimate of the four basic tastes, those qualities, to the taste of potassium chloride. And you can see that people said that was mostly salty, maybe with a little bit of bitter, but mostly salty. And if we do a magnitude estimate, so how much more salty is this, or how much of this flavor do we have in comparison to others? The sodium nitrate, that is rated as salty, sour, and bitter, and about equal components of those things. And those are much more strong tastes than sweet when we are tasting sodium nitrate. I'm going to go ahead and state right here, you do not need to know any of the chemicals that I mentioned for any of these. I was not strong in chemistry, and as far as I'm concerned, what the chemicals are telling us are about our taste receptors and our taste abilities, and that's what you want to know about, not the chemicals themselves. So as we usually do for any of our sensory modalities that we've talked about so far, we're going to talk about the sensory receptors themselves, and they are couched in, or they live in, these different areas. So we're going to start with the kind of highest level of all of this, which is the papillae. And we have different kinds of papillae all across our tongues, actually within our cheeks and on the soft palate on the top of the mouth. But the only ones that ever get talked about in any of the textbooks that I've read have been the ones that are on the tongue. And so I'm going to go through these four kinds of papillae that are on the tongue. One is filiform papillae. These are shaped like cones, and they lie all over the tongue. The fungiform papillae, which we'll spend the most time talking about most likely, are shaped like little mushrooms maybe, and they're a little bit darker red. They're found at the tip and the sides of the tongue. The foliate papillae are a series of folds at the back of the tongue, and then we also have these kind of flat mounds surrounded by trenches at the very back of the tongue, and those are the circumvallate papillae. Okay, so I'm going to talk about these papillae a bit, and we're going to start with the taste, that they are housing the taste buds and the taste cells. Although the filiform papillae, they are the only papillae that do not contain taste buds or taste cells. And so if you remember what I just said, or you look at the picture there, you'll notice that it's the filiform papillae that are all over the tongue, and in the center of the tongue, the fungiform papillae are at the tip, the front tip and the sides. The foliate and the circumvallate papillae are back there as we move towards the back of the tongue. And what happens then is we have a blind spot, a taste blind spot on our tongue, where there are filiform papillae that have no taste. But what occurs is we have mechanoreceptors, we have touch receptors surrounding those papillae, and we can still feel what's on our tongue. And so if you go and you take a little cotton ball and you soak it in some salty solution or bitter solution, whatever you want to soak it in, and then you take it and you touch it so it's just on the center of your tongue, and you're careful not to touch the tips, the side, or the back, what you'll notice is you don't taste it at all. And then if you take it out and then you move it and you touch the tip of your tongue and move it to the center of your tongue, now it feels like you're tasting that throughout your whole mouth. But what's really happening is you're tasting it on the fungiform papillae at the tips of the tongue, and then you're feeling that movement to the center of the tongue, and that the brain fills in that blind spot, that taste blind spot, just like it does for vision. Okay, for humans, the tongue contains about 10,000 taste buds and about 50 to 100 taste cells per taste bud, which means we have approximately, it's been approximated as about 50,000 taste cells. I know that doesn't really multiply out, but that's usually 50 to 100,000 is usually what I see as the approximation of how many taste cells we have. And just for a fun species comparison, I'm going to do I might be slightly off on the chickens. They either have 12 or 24 taste cells. So we have 50,000. They have at most 24. This is why you can feed chickens just about anything, and they're okay with that. They really don't care about what it tastes like. If we're going to compare ourselves to catfish, catfish have 175,000 taste cells, so many more than us, at least 3 times as many as us, and their taste cells are not only in their mouths but on the outside of their body. What they are doing is they're just picking things up. They're bottom feeders, right? They're just picking things up and basically tasting them and spitting them back out if they don't want anything to do with them. So they are the rude person at your party who is taking bites out of things, putting it back. Not just double dipping, but this is the height of being rude. But that's the catfish. Not to put down your catfish, because catfish are awesome. Anyway, so we have, if you look at the picture from Goldstein, we're looking at the tongue, and he is showing us a taste, the top of the taste bud, or at the top of the papillae, magnifying that to show us the taste bud. And so if we go down to the bottom of that garlic looking structure where we have these what look like cloves, and what's in the middle there, that purple part is showing us the soma of our taste cell. So our taste buds are made up of these many taste cells, which again kind of look like cloves of garlic coming together, or I'm sorry, yeah, cloves of garlic coming together for the garlic. And then if we pull that out, one of those taste cells out, as he does, as we go to the right, and we go up to the taste pore, or the very tip of the taste cell there, which is going into the taste pore, we go up into that, you can see going back up to the top right, the taste receptors. Okay, so those receptor sites where things that are creating sour and salty tastes, those chemicals can just pass on through. And then things that are causing, substances that are causing sweet and bitter tastes, those fit into the receptor sites. And we see those kinds of molecules like we saw, I think, interestingly, and we're going to see this again in olfaction, like we saw in the photoreceptors, those molecules that pass through the membrane 7 times and have this metabotropic effect. So we're going to, with sweet and bitter tastes, we're going to have to influence that whole taste cell. Transduction occurs when those chemicals come into contact with the receptor sites at the tip of those taste cells, in that little taste pore, those cilia that are poking up in there. And I'm going to do my word art, which I hope I get to keep. We'll see. It causes some problems for both saving and sometimes my voiceovers, but it's transduction. I mean, this is the magical thing that happens that allows us to taste these basic taste qualities. And again, I'm showing the 4 basic ones that have been known throughout much of our history of research. I am including information here about a number of cranial nerves that allow for transmission from those electrical signals from the tongue to the brain. The only one we're going to continue talking about, and the only one you need to know, is the chorda tympani. Okay, but I'm going to go ahead and talk about all of them. The chorda tympani takes information from the taste cells on the front and the sides of the tongue to the brain. We're going to see this all synapse in the medulla. The glossopharyngeal nerve takes information from the back of the tongue to the brain, the vagus nerve from the mouth and throat, and the superficial petronasal, that cranial nerve, comes from the soft palate and the top of the mouth to give us information about taste that is experienced up there. Again, we're only going to keep talking about the chorda tympani, and it's the only one you need to know. You can go ahead and notice, if you will, that the vagus nerve, I talked about that a bit with touch and the importance of the vagus nerve for when we are touched, and that is activating the parasympathetic nervous system and allowing us to take in nutrients. But there it is also involved in the sense of taste taste coming from the mouth and throat. But we'll only keep talking about the chorda tympani. It's the only one you need to know. So nerve fibers from the tongue, mouth, and throat make connections in the medulla at the base of the brain or in the brainstem, and the specific area of the medulla where they all synapse is called the nucleus of the solitary tract. And there's a picture of it there showing that first little area in the medulla, the nucleus of the solitary tract. That information goes from the medulla, so our first area in the brain where it synapses, to guess where. It should not be a surprise for you. It goes to the thalamus, as so far all of our sensory information has gone through the thalamus. Okay, it goes to the thalamus, and from the thalamus it goes to the insula, where we have our primary taste receiving area, and to the frontal operculum cortex. These are actually both considered primary taste receiving areas, but often the insula is where many of my textbooks put this. Whether or not, okay, this is in the temporal lobe, the frontal lobe, or the parietal lobe is different in different textbooks, as this is really where those lobes come together. And from there, from those really receiving areas, it goes to the orbitofrontal cortex. So far, we have sent our information for vision and for hearing and for our feeling our body. That information is really towards the back of the brain, and it is going to go into some association areas before going into the prefrontal cortex. But this goes straight from the primary receiving area, this insula, and the frontal operculum cortex to the orbitofrontal cortex. And this is called the orbitofrontal cortex as it is the frontal area, the part of the frontal lobes that is just above the orbits of the eyes. So that if you can just kind of touch the top of your brain there near your eyes, that is where we're going to put together the information from taste and smell so that we experience flavor. But there's also right around there areas where we have a lot of emotional processing of especially inhibiting potential of emotions that might take over, and our feelings of guilt, and a lot of our decision making. A lot of that is up in our prefrontal cortex, just right around this orbitofrontal cortex, where we're going to put together information from taste and smell and touch and sight and hearing so that we can experience our dining, our flavor, and our experience of food.